Hello, my name is Miss Maycutt and I teach environmental science at Ballard High School, and I'm here to present the Acid Deposition Lab Home Challenge. And the reason I decided to take this on is because all of our labs, it's so hard being at home to do some of the cool labs that we get to usually do at school. So I took this one and adapted it so you can pull it off using the regular equipment and materials that you have in your kitchen. All right, so acid deposition, this is part of our air pollution unit. We've been studying acid rain, essential knowledge 7.7. We'll also get into LD50, which is EK 8.12 and 8.13 for those of you that are in AP environmental science. And the general concepts being is that we get pollutants, we get NOx and SOx into our atmosphere. They combine with water and oxygen and produce sulfuric acid, nitric acid that then descend upon our terrestrial and aquatic landscapes, ecosystems. So what happens? How does that impact them? Well, let's find out. So to do that, this will be our procedures. You also have a set of materials. For the materials, you will need to have some jar containers to put the concentrations of acid into. You will need vinegar. That is our acid of choice. I recommend white vinegar or apple cider vinegar, as the other ones often have sugar. You'll need some seeds. Radish, peas, and beans work best because they have a nice, quick germination. You can do just 5. We're gonna have, as you can see here, we're gonna have 5 different concentrations. You can do 5 beans, seeds of each, or you can do 10. We will be doing the LD50, so having 10 is kind of convenient. Or you can just do an even number. I'll need paper towels, vinegar, masking tape, some pens. What am I missing? Oh, and make sure that your water is either spring water or take your tap water, let it sit out for 24 hours to remove some of the chlorine. All right. When we're all done, you'll also need a ruler that has millimeters on it so you can measure your plant's growth. Okay, so that's the basic materials. And then to set it up, the biggest challenge is doing the cereal dilution. So I'll walk you through the steps. Label ahead of time, right? It's just, there you're ready to go. So label your plates, label your cups. Label them with 100%, 10, 1.1, and 0. That's the labeling. Set up your plates and lids and get your seeds out. Count out however many you're going to put in each of the dishes. Now for containers, instead of a Petri dish, that's what we usually use. These are my kids' little snack bowls, right? So this is my 100%, 100, 10, and so forth. You also want to take some paper towel. So I took one paper towel, folded it twice, and then cut it to fit. When you put the seeds in, you'll want to take one layer, just grab the top layer, and put them underneath that because, of course, these don't have lids. You need air circulation, but you don't want them to dry out. So have another piece to put on top, and there you go. You're set up with your little dish. Use, of course, the same amount of paper towel in every dish, so you're controlling your variables. All right. Then I chose jar containers. They need to hold at least a cup. They can be of various sizes. You'll measure how much goes into them. And then label them. I colored the 100% so then you'll be able to see the cereal dilution when we do that. And you'll need a measuring cup and measuring spoons. So the trickiest thing is the cereal dilution. And for that, we'll use our spoons and our measuring cup. Now, usually we're using milliliters, but I don't have a graduated cylinder around here. So what I did is there's 48 teaspoons in 1 cup. So I rounded it a little bit. So we'll be using 5 teaspoons, right? We'll be removing 5 teaspoons, which will give us 10% of 1 cup. So 50 is a cup, so 5 is 10%. So you start with 1 cup of vinegar in the first one. And you can actually go ahead and do the last one too, and just make that water. That'll be the ends. The tricky part are the ones in between. So fill this with 1 cup of water. So fill the second cup with 1 cup of water. Then you're going to remove 5 teaspoons. 5 teaspoons are going to go away. And then you're going to add 5 teaspoons of pure vinegar into there, right? We're going to do that for each set. So then we'll remove 5. So remove 5 again from the next one. So take 5 teaspoons out of the cup of water, and then you're going to add 5 teaspoons of the 10% to the next one. One more time. Remove 5 teaspoons, and then add 5 to the next. And that gives you cereal dilution. And when you then add it to your dishes, you'll just take your measuring cup, spoon, add a little to each of your plates. Make sure it's the same quantity. So if it's a small dish, you'll just add 1 teaspoon. So you can use lids, yogurt lids, or any kind of flat. It just needs to be flat. That's the main thing. So any kind of lid. This I just would add 1 teaspoon. Here you might add 2 teaspoons. It's a little bigger. Make sure as you keep adding water, you're consistent doing that to all of the dishes at the same time. And then you watch for growth. When you finally have got some good growth, it'll take a week to 2 weeks, maybe 3. When you finally get your little plant growth, you'll measure that, just get it in millimeters. All right, so measure in millimeters, just use your basic ruler. And record that. You'll need a method of data collection. So set it up something like this, where you've got each of your percent cereal dilutions, and then you'll have each of your seeds total samples, get their totals, their total lengths, find their average, and then you can plot that. Now, once you set this up, don't forget at some point, make a prediction. This is an experiment, right? What do you think is going to happen? Where do you think you're going to, the 50% lethal dose will occur? So at what concentration, what dilution, are you going to be losing 50% of your seeds? All right, that's the challenge. Do it at home, have some fun, and thanks for joining me for science. Here's the final setup. Have fun with your experiment.